Can I give it to you later? No. Just 1 second, Alwyn. Here. Turn this off. Maybe that's for sound. Yeah. Stella, can you bring me a chair, please? But come on to the other side, because then it's. All right, Owen. That's better.
Yep.
You want to try this, the clicker? Oh, it's connected now? Just press the button. Yeah. All right, go back, Alwyn. Yeah?
Yeah.
Oh. Okay. No. Try it again? Yes.
So yeah.
And speak a little bit louder.
Okay, I'll try.
Projecting, not louder.
I hope we changed the setup here a little bit, so I hope it's better. It's easier to understand through Zoom. Also try to speak up a little bit. This session about the workshop is like what we really want to talk about is the introduction on Diadema restoration and different approaches that are existing or that we developed and are now existing to restore these important herbivores. So we'll very shortly touch upon like why do we actually want recovery of Diadema populations, what is currently constraining natural recovery, and what are the restoration approaches that we have available so far and that we can use. So as I told earlier, the die-off in the 80s resulted in a 98% of population reduction. And after the die-off, the changes on the Caribbean coral reefs became clear very soon in terms that turf algae started to increase and microalgae started to increase, and in some cases even overgrew corals and prevent coral recruitment. There has been some recovery, mostly at sheltered at shallow locations. And in 2015, they estimated that the current population densities are around 12% of what they were before the die-off. On the places that are actually recovered, there was a phase shift reversal recorded. So there was less microalgae, or microalgae cover decreased. There was a higher coral recruit density, and those coral recruits also grew faster and had a higher chance to survive. And in the end, also cover increased. So all very good developments. And this picture from Jadi et al. 2010 shows it really clearly. On the top of the picture, you see the Diadema zone, the urchin zone, where there's a lot of Diadema around. And you don't see so many algae. And in the bottom part of the picture, you see the algae zone. There's no Diadema, there's just Dictyota, outgrown, everything. So this is also where the recorded higher coral recruit densities and growth and survival in the urchin zone compared to the algae zone. So that's why we want recovery. We want more areas with a lot of coral recruitment and survival and growth, of course. But so far there has been little recovery on most of the reefs, and especially deeper reefs have almost no Diadema in most places of the Caribbean, although there's some exceptions. So why does it take so long for recovery to take place? I mean, we're almost 4 decades since the die-off. and we still won't have a full recovery. Well, there's different hypotheses about that, and I'm going to go through them quickly. There's indications in some places for an LE effect, so that means that there's not enough adults to result in successful fertilization. Also, there's fewer source populations altogether, so some populations just have been wiped out, and they do not contribute anymore to the larvae pool. So if a certain reef doesn't have an upstream population anymore, there's also no larvae going together, and there's no chance of recruitment. There's a big habitat change that happened after the die-off. So most reefs are now turf and microalgae dominated instead of coral dominated and CCA dominated. So the dominant benthic group changed. And this has implications for the Diadema. It means less suitable settlement substrate available, less available food for settlers, and also more habitat for micropredators. And also the habitat changed in the sense that less coral also means less shelter. So shelter availability in general of Caribbean coral reef has gone down since the 80s. So that can also limit the recovery. And so this is all these different factors, they interact with the life cycle of Diadema. So here we have the adults in the bottom corner here, and this one, and if eggs and sperm leave in the water column, fertilization takes place. And we have a larvae, or the fertilized egg can develop into a larvae, and then dispersion takes place to other reefs. They're in the water column for 30 till sometimes 90 days. And in that time, the larvae becomes competent. That means ready to go through metamorphosis. And then settlement takes place. And we have a settler, and you actually never observe settlers in the reefs. They're very small. You immediately start hiding in the rocks or into cracks, and you never really see them, and they're super small. But if they survive this period, if there's post-settlement survival, then a recruit can become visible. And a recruit is around 5 millimeter big, the test size, so the body size. And those are the first urchins you observe on the reef, usually. And then if the recruit survives, you have another adult. And then the life cycle closes. But unfortunately, there's all these factors that constrain recovery. And I already talked about the low adult densities that can limit fertilization. There's also the problem of fewer source populations than before the die-off, and that limits dispersion to certain reefs that don't have an upstream population anymore. And there's this big habitat change from coral CCA dominated towards turf and microalgae dominated. And that has a serious problems in many different ways. First, especially turf algae seem to hamper settlements. So settlers really like relatively clean substrate with a fresh biofilm, and they also like CCA. Tom is going to tell you more about that later. And they don't really like turf algae so much, and about microalgae we're not really sure yet. But the amount of suitable settlement substrate has declined, very likely. Also, turf and microalgae are not the right food for settlers. They feed on a fresh biofilm the first couple of weeks, and that's less available, and that can reduce their survival. So that's this other arrow. And then turf and microalgae are the perfect habitats, not for Diadema, but for the predators. So micropredators like crabs, like fireworms, they like to hide in the microalgae. And that, of course, also has a negative effect on the post-settlement survival. So there's another arrow there. And then last, there's some indications that certain types of macroalgae and also turf algae can fill certain crevices and cracks, actually reducing the shelter availability. So there's a negative effect of turf and macroalgae on the shelter availability for Diadema. Shelter availability on itself is, of course, a good thing, can facilitate post-settlement survival and post-recruit survival, but shelter in general declined in the Caribbean over the last few decades. And then lastly, we have to recruit adult predator, for example, this queen triggerfish, there's many more, and they also have a negative effect on the post-recruit survival. So all these different effects, all these different factors affect the recovery, and depending on your location, one factor can have a bigger effect than the other. There's one arrow I would say missing here, and that is this one. That's the effect that the adults have on the habitat. And that's a very important one because there's no adults, also this habitat doesn't change. As soon as you have the adults, you have your clean surface with a fresh biofilm, you have your CCA, you don't have the hiding spaces for the micropredators anymore because the macroalgae are gone. And then recovery starts to take place. So in places where we see the adults, like small recovered populations, we also see a lot of juveniles because of this habitat change. So you could say that because there's no adults, it stays in this negative feedback loop, and we have to, we need some sort of intervention to come from this negative feedback loop in a positive feedback loop in which adult Diadema can once again promote settlement of new Diadema and get a healthy population going. So Diadema restoration is one of the things that can be implemented locally. So there's many things that threaten our reefs, but a lot of those things have to be targeted like regionally or internationally. But this is one of the things you can do relatively locally. It has a large impact, facilitate natural coral recruitment, which is of course very important, but there's also indications, and we just heard some presented by Kelly actually, that they can also increase the survival of coral outlands, which is of course also very important. And especially in reefs like we have here on Saba that are overgrown by macroalgae, you can ask yourself the question, does it even make sense to restore corals if they're overgrown with macroalgae again? So we need to do something about those macroalgae at the same time we restore corals. So there's a couple different restoration approaches that are available. You can translocate animals from one site to another. So you can use a recovered population, if it's big enough and maybe resource limited, to restore another area. You can collect settlers in the wild, take them to the lab, take them to a grow-out system on land and restore them when they're big enough. You can also culture them from gametes. And all these different methods rely on restocking. So you basically produce individuals in a certain way, or you take them from the reef and then put them on another location. And then we have a fourth approach, which we term assisted natural recovery, in which you provide the settlement substrate on the reef, and you don't touch the Diadema, and still it can result in a recovered population. And of course, there's always future approaches. So we'll go through these different techniques to get an idea what's suitable for which reef or for which island. So first of all, translocation, which is probably the easiest to start with, but also very limited in its scale, because you need at least hundreds of excess individuals. That means that you basically need a population that cannot expand by itself anymore, because the whole breakwater is, for example, full of Diadema, and there's no food for them to expand. Then you could consider such a thing, or maybe if the donor site is going to be destroyed by coastal development, dredging, of course, then you can use it too. Certain advantages and disadvantages, well, it's for sure not for large-scale application. We don't have enough Diadema, and it's also very dependent on the natural populations. And at the moment, it's not going so well with the natural populations. Just to make sure, it's not a good time altogether to move Diadema from one place to the other right now, because you might just spread a disease that you're not aware of yet. Then collecting settlers and head starting them on land in a land-based facility. This method is developed by Stacey Williams from ICARIB, and she also helped us to establish it here on Saba. So thanks a lot, Stacey, for that. I'm not sure if you're listening, but I definitely want to give the credits of this method to you. She also described it in a couple of publications, and basically what you have to do first is determine the settlement hotspots around your island, because you need to collect a lot of settlers. You also have to determine the settlement season, and it seems to be that throughout the Caribbean, the summer months are where settlements takes place, but it might differ a bit per region where in the summer it happens. So you definitely have to find that out if that not has been done before. And then you place settlement collectors mid-water, and it can be all kinds of different collectors. You see a frame of bioballs here. I also brought some bioballs to inspect for you later. These are plastic balls that are used in aquaculture filters. They have a lot of surface area, and the Diadema really like to settle on them. But you can use all different kinds of materials, and I would say basically Diadema settle on anything that's in a water column, if it's there in the right time of the year. So you place them mid-water because that removes the predation pressure. And you replace them monthly during the settlement season. And by replacing them monthly, you keep this fresh biofilm on the settlement collectors. And that's really important because that's a settlement view. And we also tried leaving collectors longer in the water, and then they just get overgrown with all kinds of algae mostly. And they face the same problems as the natural reefs, and they don't have as many settlers anymore. Also because micropredators start to settle on the streamers and probably eat the Diadema. I need to click on the screen to move on. So I'm going to present some of the data we gathered around St. Eustatius and Saba. This is around Statia. We sampled the 5 locations every 2 weeks with different collectors, and to determine the best type of collector, the best material, most optimal, and also to get a feeling for the settlement season. And what you can see here, the months are in the bottom of the screen, and might not be clear to everyone, but there's a peak in June, settlement peak in June. Then there's not so much settlement in July on most locations, and there's another peak in August. And then the rest of the year, settlement is pretty low, but those peaks, on the other hand, are very high. And they are, in terms of like individuals per square meter, they're comparable to pre-die-off densities measured on Curacao. Like pre-die-off density, settlement densities around Statia and St. Eustatius have never been determined, so we don't really know. But on Curacao, they really did a great job there. And these peak settlement rates are comparable to their pre-die-off settlement rates. Around Saba, we chose 9 different locations, and we followed, we measured settlement for a year. And there's a very big distinction. So this shows how important it is to determine your settlement hotspots. Because you're in the wrong location, for example, these red crosses over here, those are Cove Bay and Cork, 2 locations here that almost had no settlement whatsoever throughout the year. So we found very few of them. And then the green crosses over here, they had very high settlement rates, and they got as much as 1,500 individuals per square meter per year. So there's a lot of individuals to be gathered on those locations, and all the other locations have more or less intermediate settlement densities. So these were the different settlement collectors that we compared. We did this experiment on Statia, St. Eustatius, and we used settlement panels. Those are also described by Bach in a paper published in 1984, and by Vermeij, a paper published in 2010. And we wanted to use those so we could properly compare settlement rates with the settlement rates that they measured. And then we also included a range of mid-water collectors. So we used artificial turf, we used straight rope. Straight rope is used as a mussel seed collection device in the Netherlands. So that's how we came to use straight rope. It also provides a lot of surface, actually has the highest surface of all these different materials. We used a doormat material. That's what Stacy mostly used in Puerto Rico. And we used these bioballs because we knew they had a lot of surface area, and we thought they might work well. And then we compared all of them. And one of the things that were most interesting is that the panel didn't work as well. We didn't collect as many Diadema on the panels compared to the mid-water devices. And it might have to do with multiple things. There's probably more predation on the panels than on the mid-water devices. Also, the Diadema that are already in the bottom water layer have a lot of other settlement substrate because they have all the reef around there, so they might settle on that instead of or panels. So there's different explanations for that. But if your purpose is to get a feeling of settlement or gather as much Diadema as you want, I would definitely recommend to use mid-water collectors. And then the freight rope didn't work as well. We also collected mostly silt with it. So it took a lot of time to to clean them and to rinse them out. But the doormat, artificial turf, and bioballs all did very well. And in the end, we decided to continue with the bioballs for several reasons. They don't collect as much sediment, so they're pretty easy to rinse. They're also relatively cheap, and they don't break off. So that's one of the problems we had with the artificial turf, is that pieces start to come loose and they pollute the environment. And of course, we don't want that. So that's why we choose to continue with the bioballs from this experiment onwards. So this is a close-up of the bioballs. Might be useful for those on Zoom to see it a little bit more closely, but they have a lot of surface area. We also did some follow-up experiments. What is the best bioball configuration? Like, you can put them all together in a net. So that's what we did, 50 bioballs in a net. And we also did 50 bioballs on a string. So then it looks like this. And we looked at the amount of settlers we collected per bioball. And then the strings are much more effective than the balls. And it's also pretty logical, because the bioballs in the middle of the net, they don't receive any diadema, because those already settled on the outside balls. It's still useful to know. So these are the balls, the clumps of bioballs. So some conclusions of this settlement study. Peak settlement rates were similar to pre-dive rates measured on Curaçao. Settlement over the year is still lower. And bioballs are most effective in collecting settlers. They were slightly more effective than the other materials, and also most consistent. I forgot to tell that. So that's also a good reason to use them. And strings of bioballs works better than clumps of bioballs. And then there's a short movie here. If someone can press the movie, please, because this is what we're after. Very small diadema settlers. This one is almost a millimeter big, but they can be as small as 0.4 millimeters. So we're looking at very small individuals. So then, in the workshop this afternoon, all of you can experience the bioballs, and yeah, if it's feasible for your island, because we deployed a couple a month ago and retrieved them yesterday, so we can see if there's any diadema in there. If you have those settlers, of course, they're not ready yet to put them on the reef. They might be ready to put them, to co-culture them with corals, like you suggested. That's a very good approach. But we want to get them bigger, and we use this grow-out systems that are actually super easy. They're standalone. We do a weekly water exchange. They have air-driven pump filters to make sure nothing mechanical is happening in there, so they don't get into a filter and die. They feed on the biofilm for the first 3 weeks, so you have to make sure that your tank is properly in balance before you put them in there. There's a little bit of biofilm for them to eat. And we feed the juveniles mostly on macroalgae. We give them a 12-hour light, 12-hour dark regime, and that also promotes some algae growth in the tanks that they can feed from. So this is how easy it can be. The settlers are between 0.4 millimeter and 2 millimeter usually, and in 6 months they can grow out to juveniles of around 2 to 3 centimeters. And they might be ready to put on the reef, depending on how big you want them. We have very varying survival rates, and that's really dependent for us on your initial stocking density and how careful we were with removing micropredators. So we had 30 to 70% survival, and the survival seems to be dependent on the size in which we collect them. Like, bigger settlers have a higher chance of survival. We never seem to get more than around 50 per tank. Those tanks are approximately 60, 70 liters. And we tried stocking them with 200 individuals, and we still got 500 recruits from them. So density seems to be important. And we have to inspect them for micropredators that enter the tanks with the water, or with, especially with the seaweed. For example, as eggs are laid on the seaweed, and we introduce crabs and fireworms. And if we leave them in the tanks, they can kill off a whole tank in like a couple of days. So we have to check for micropredators at least weekly and remove all the small crabs and whatever is in them. And that's also the reason why we keep our tanks relatively clean, and we don't put too many rocks in there, especially at the beginning, because we want to keep those micropredators under control. So some conclusions of this approach, like collecting the settlers, head starting them, and reintroducing them. I would say it's pretty low tech and also low cost. You can probably start doing this with as little investment as $1,000. First, it's pretty important to identify your settlement hotspot because that's where you have to collect the settlers, and then identify the settlement season as well because you have to know in which months you are going to deploy the settlement collectors. Of course, you're dependent on the natural reproductive season, so usually the summer months. And you're also dependent on source population, and this might or might not, we don't really know, become a problem after this die-off went around in some of the Caribbean islands. Oh, I also put a note there, the settlement might be less seasonal on Bonaire, Curaçao, and Aruba, especially the observations from that paper from Buck in 1984 show that there's actually settlement year-round. And we don't really have that here. And I'm not sure what happened after the die-off, if it's still as year-round as before, but that might be something interesting to look into. So then we have the approach culture from gametes, which is usually seen as pretty difficult, but by now we're able to conclude that's certainly possible. We developed a new shaker table method in which we, on a small scale, can produce settlers with every cohort. And Tom, yeah, he's the one that deserves credits for this new method, and he's going to tell you all about it in the next session that will also be available on Zoom through a different link. And then I want to step back a little bit before I introduce the next approach in this limited natural recovery. Because with our settlement studies around Saba and St. Eustatius, we can actually conclude that peak settlement rates are similar to pre-dive rates measured on Curaçao, and also that there's potential for natural recovery. So apparently, it's not so much the LE effect that we're talking about, the low adult densities, or the few source populations that are the problems, at least not at some of our reefs, but it's more the post-settlement survival and the post-recruit survival, and also the settlement bottleneck here that might be the problems. So we decided to work on a different approach, and that's approach we term assisted natural recovery, and it's already used in forest restoration. And the aim of this method is basically not restore the trees themselves, but see what is in the way of natural recovery. So, for example, can we transplant a seed bank, or can we stabilize the bottom, or remove other disturbances, and then let the forest recover itself? And we wanted to do the same with the Diadema. Say the problem is there's not enough settlement substrate. Can we not just provide them with suitable settlement substrate and then let nature take care of the rest? Because all these different approaches are still pretty time and resource consuming. So what we did, we chose a location with very high settlement. We selected 6 similar patch reefs, and 3 of them got these bioball streamers attached directly to the reef. And of course, 3 of them did not get any treatment, and those were all control reefs. And we deployed the bioball streamers just at the onset of the settlement season, so they would have a fresh biofilm layer, which the larvae like if they're ready to settle. We also deployed proxy streamers, not on the reef, but on cinder blocks next to the reef. And those were replaced monthly to get a good idea of the actual settlement. But the streamers on the reef, we of course left in place. And the hypothesis was that the Diadema would settle on them and then outgrow the spaces on the bioball and move to the reef. And yeah, we hope to start population recovery with doing that. So the first step was settlement, and that worked really well actually. These are the amount of settlers per streamer. And in May, we had around 10. And in June, we had 15 per streamer. So that's a couple hundred per reef. And then settlement decreased again. And in July till October, we didn't have as many settlers on the streamers. So we're mostly dependent on this initial settlement. And this is a picture of one of the settlers on the bioball on the reef. So we actually observed them settling on them, and there was quite a few of them. So then the settlers had to outgrow their safe space on the bioballs and move to the reefs or follow the bioballs on the reef. And there we could measure them as recruits, in which the urchins are around 5 millimeter big. That's first when you start to observe them. And we counted, every month we counted the amount of Diadema recruits on the reefs. And we started with 0 because there were no Diadema on those reefs. And then on both reefs, actually the amount of settlers went up to on average 2 Diadema per reef. But then it started to decrease again for the control reef, while the treatment reefs kept increasing. In the last few months, we had significantly more Diadema on the treatment reefs compared to the control reefs. So definitely something was going on there, although we didn't manage to really establish a population last year. And that might have to do with a relatively low post-settlement survival. Remember I told you we had a couple hundred settlers on the proxy streamers. Those were probably also the numbers we had on the streamers attached to the reefs. But still, we only measured as much as 15 Diadema on a single reef recruits. So there's quite a gap there between amount of settlers and the amount of recruits. So there's still a lot of post-settlement mortality going on, which might actually be pretty normal. But also another thing, and you can see it in the size distribution here at the bottom of the sheet. This is the size distribution for July, which is around, most of the Diadema are between 10 and 20 millimeter. And then you see the size distribution for the end of the experiment, November, and still they didn't become any bigger than 20 millimeter. So, and they actually grow 3 to 6 millimeter per month. So these are definitely different Diadema that we're measuring here. So we have continued recruitment on those reefs, but then when they reach a certain time, reach a certain size, they disappear again. And of course, they can move to a different location, but we also inspected the reefs around the patch reefs. We didn't find any. So this is most likely predation, because we saw individuals in the same place, in the same shelter, a couple months after each other. They looked healthy, they were growing, and then they disappeared. So probably predation, but we're not sure yet. So then we're back to this overview, and we can conclude that actually here the recruit adult predator also has an effect on the population establishment, and that also has been shown before by others and by modeling studies. Especially if there's only a few that make it to the recruit stage, it's pretty easy to pick them off. And if, for example, queen triggerfish eats 2 every day, that's enough to prevent big population recovery. So a couple of conclusions from this assisted natural recovery method. It's low cost and low tech, and investment of equipment is even lower than with collecting settlers, a couple $100. I think it has potential for large-scale application because it's not very time consuming, but it needs optimization, of course. Before we can do it large scale, we have to show that it works. So we thought about different things to increase post-settlement survival. One of those is use bioballs with larger shelter spaces, so they can stay mid-water longer before they move to the reef, and then they can skip this vulnerable phase in which they are as a settler on the reef, and also prepare the habitat by removing macroalgae, and by doing that, removing a lot of the micro predators. And that can be done by restocking Diadema, adult Diadema at the same time. It can also be done by brushing the reef with iron brushes or steel brushes, or even by restoring different herbivores that can also do this job. And this method, of course, is of course also dependent on natural settlement. And then this is the BioBall 2.0 that we termed the Diadema booster. I actually have it here. So it has the same small settlement spaces as the initial BioBall in the middle, but also has, if they outgrow that, they have larger shelter spaces on the side, in which we think they can live till they're around 1.5 to 2 centimeter. And if they move to the reef, then they're less vulnerable. And also they might do it in larger numbers, so they actually, the recruit adult predators are not such a big problem anymore. This new bioball, we hope, is also suitable for the collection of juveniles. I mean, if you want juveniles of 1 to 2 centimeters anyway, it would be much easier to collect them at that size than to collect them at 0.4 millimeter. So that's one of the things we hope to try this year, to see if we can collect larger juveniles with this new type of bioball that we developed. Second?
They still need water, but they're bigger.
Yeah.
We still need water.
Yeah, exactly. So we still plan to deploy them mid-water. Yeah.
For the bioballs, take them out every one.
No. So the question is, do you reuse the bioballs? And of course, we reuse them as much as possible. That's also, yeah, that works really well. Only these bigger bioballs, we don't plan to take them out every month because we want them to grow bigger. So we put them in and then we remove them after 4 months, for example, see how big the juveniles are, and then we can use them again next year. That's the plan. So some conclusions about the different restoration approaches that we now discussed. Translocation can only be done on a relatively small scale, very dependent on natural populations. Settler collection is low tech and low cost, relatively easy if you get a feeling with it. Also dependent on natural reproduction. Well, Tom is going to tell you more about culture from gametes, so I will wait with any conclusions for that. And assisted natural recovery is, yeah, shows hopeful results, but needs optimization. And of course, it's still dependent on natural reproduction events and natural source populations. I want to thank a lot of different organizations, all our partners in the Diadema project, all the people that helped us, advised us, and also all the students that did their internship or thesis with us and helped us by doing that. Thank you also for listening, and of course I'm open for any questions.
Before you start, we have about 10 minutes before this session. And we also have some questions, most likely. So maybe just 2 for now for the group, and then after you can discuss during lunchtime your questions.
Yeah.
Sure, when you mentioned it, but I missed it. What are the bioballs made out of material?
Yeah, it's a polyethylene. It's a plastic. So the bioballs are made from polyethylene. Any other questions for now?
There's a question here.
Yeah, sure, yeah.
It's from Joe, the University of Miami. Can you please describe how you get the diadema from the bioballs to the tanks? Presumably, this is a step in the process where they may be more likely to fall off or get crushed.
Yeah. So
if you could read it, also it's right there.
Oh, yeah. So this is a very good question. Like, how do we get them from the bioballs to the tanks? What we do, we collect the bioballs underwater with Ziploc bags. So we put them in there and make sure no diadema fall off, because sometimes they fall off pretty easily. We take them all to the lab, and there we rinse out every string of bioballs in different containers. And what we usually do is we take 5 white containers, put them next to each other, and rinse gently in the beginning and rinse them more roughly towards the last container. And then we found out that we get almost all of them off. Of course, you don't immediately see them because they're in between all the debris that's coming off and other animals. But the nice thing about the Diadema settlers is that they adhere to the surface of the containers very quickly. So if you gently tilt the container a little bit and you start to see all the sediment moving up and down, and you have to train your eye a little bit for it, but then you see a couple spots that are staying in one position, that's the Diadema. Of course, they're also very red, bright red, so that's also another point to identify them. We take them out of the container. Usually I just do it with my hands. You can also use pipette, put them in a separate cup or whatever, and then put them in the settlement containers. So that's how we do that step. And for the people here, we're also going to practice that this afternoon. It's one of the parallel sessions that we're going to do together. So.
Sorry? You don't know if there's not.
Of course there will be. No, it's a little bit early in the settlement season, so we don't know yet. That's true. Any other questions?
If you leave the bivalves in the water, the main cause of mortality was the turf algae growing or predation. That's why you want to bring in the lab. So I think the lab staff going to reef phase is definitely a limit in terms of scalability, right?
Yep.
And trying to keep them more in the field and the hiccup or in the bottleneck was the fact that predation and algae growth is the main stressors.
Yeah. Let me repeat that question. So, can you not leave the bioballs longer in the field and culture them shorter in the lab because that's the most resource? Very good question. So the problems with these initial bioballs is that they outgrow the shelter within a month or two. That's also when we retrieve them already. So you cannot leave. You could leave them maybe for 2 months, and then you get slightly bigger individuals, but not longer than that because then they outgrow the bioball and just fall on the sand or on the reef down there. So that's actually what we try to use that with the assisted natural recovery approach. And that's also the reason we developed this new Diadema booster with larger shelter spaces. So we can, the idea is to keep this in the water for a couple months and then get juveniles of 1 to 2 centimeters that can immediately be transplanted to the reef.
We have a question here.
Yep.
From Karen from Saba. Your advice for finding settlement hotspots, near adults, shallows, current, no current?
Yeah, that's also a very good question. Like, should we, so if we want to find settlement hotspots, should we go towards like current populations because apparently they're settling there? Not necessarily, to be honest. Like if we, that's what we first did. And those are not the places with the highest settlement. There's settlement there for sure. But there's other places that have much more settlement, and those are not the places that have necessarily adult populations. So I would just see what's feasible, see what are dive locations that you go to regularly because there's coral nurseries or whatsoever, and just deploy a couple lines. 10 would be great, for example, and then you replace them, or 10 locations would be great, and you replace the collectors monthly during the summer months, maybe only 3 or 4 months, and you get a pretty good feeling which sites have settlement, a lot of settlement or no settlement.
Great, thanks, Owen.
Okay.
For now, so we'll end this session and maybe take a quick break, get some drinks, use the restroom while I get sorted with the next session with Tom.